Welcome, everybody. I'm Steve Johns. This is a presentation for my Chem 530 course at GCU. And it's going to be about ion channels and neuron communication, sort of an overview of these. Now, with ion channels, they are protein channels, chains of amino acids that are integral proteins that cross these phospholipid bilayers. And there's different kinds. We've got ligand-gated ion channels. These are transmembrane proteins that cross that membrane. And then what they do is when a particular ligand there, or like a neurotransmitter, some sort of receptor site where something binds to send a signal to open that channel, it opens up and things can pass freely through that. It can be a specific molecule down a concentration gradient passively. An example of these would be neurotransmitters. Now, another type of ion channel would be a mechanically gated one. This is one where the gate is activated based on some sort of physical change. It could be a pressure or some kind of vibration. An example of this is touch, such as the skin. And then we also have ion channels that are always open, such as, so these ones that are always open allow things to openly pass down a concentration gradient across it. So examples of this would be like the potassium leak channel. And then we also have voltage gated ones. These are a little bit different. These are where the transmembrane protein, there's some kind of ion that crosses through this, where the opening and closings are regulated by a particular membrane potential, meaning a charge difference across it, driving it. So fundamentally, with transport across these membranes, we have two different kinds. We have transports that are passive, and the most fundamental passive transport is simply diffusion, where really small molecules or small uncharged molecules like carbon dioxide or oxygen, they're able to diffuse down a concentration gradient from an area of high concentration to an area of low. Now, for molecules that are too big to cross that membrane, or they may be charged, they're unable to cross through this phospholipid bilayer. So they need some sort of gateway or channel, a passageway really, to cross that membrane. So those kinds we call facilitated diffusion. It's still diffusion where it's going from an area of high concentration to an area of low. The difference is that they need some sort of passageway for it to pass through. Now, these can let them free flow, or it can regulate the diffusion, but all of these are passive transports. Where active transport is one where energy is used to take ions, charged particles, or molecules and move them against a concentration gradient from an area of low concentration in the direction of a high. So with this, we're going to spend a little more time just talking about active transport. Active transport, it uses energy. It uses energy to move some kind of solute against a concentration gradient from an area of low to an area of high. And where facilitated diffusion really speeds up the transport of a solute by providing it really an effective passageway through that membrane, but it doesn't really alter the direction of the transport. The transport's being driven by a concentration gradient. In this case, with some of these, what we end up with is where we are able to move the substrate against that concentration gradient. So active transport allows a cell to really maintain the concentration of a gradient, keeping one side higher and one side lower, and really one that's different than the surroundings. So the sodium-potassium pump is one of these classic ones. So generically showing you an ion with a binding site, ATP is delivered and it drives that ion across. Energy is required to pull those ions across that concentration gradient. And to spend a little bit more time on it as this animation rolls through it, I'm going to walk us through the steps that occurs in the sodium-potassium pump. This specific quick case, part one here is really the sodium binding. So this particular transmembrane protein, it has a binding site. where sodium that's in lower concentration inside the cell in the cytoplasm, there is a binding site that's preferential to sodium. So once those three are bound there, we move into the second stage where an ATP binds to a binding site of the protein where it's phosphorylated, where a phosphate is removed, and we end up with ADP. And with that, the shape of the protein changes, where then those three sodiums are expelled extracellularly. And now we move into a stage where there's a preferred binding site here that potassium can bind to. So when those two are bound to those binding sites, that phosphate is released to be an inorganic phosphate floating in the cytosol, in the cytoplasm, where then the protein's shape changes again, and those those potassium are released intracellularly. So this is where we're going from an area of low concentration to an area of high for sodium, and an area of low concentration of potassium to an area of high, where energy is being used. So here in this model over here, we have an ATP binding site here with the three sites for the sodium, where that energy is discharged, the sodium is released, potassium binds, the inorganic phosphate's released, sodium is released intracellularly, and the process continues over and over and over again. It's moving charged ions across that concentration gradient from an area of low to an area of high. So there are other methods of crossing membranes. A proton pump is another example. So a proton pump, what it does is it helps really maintain what's called a membrane potential. So a membrane potential is a voltage that occurs across a membrane. It has to do with the difference in the distribution of charged ions on one side versus the other. And really there's two combined forces collectively called the electrochemical gradient, where they really drive the diffusion of these ions, these hydrogen ions, across this membrane. So you've got a chemical force and you have an electrical force that's driving these, and referring to this as an electrogenic pump. What it's doing is it'll transport these protein, or it's a transport protein that's going to generate this voltage that's going to draw these positively charged ions towards this negative charge, and energy will be used either from ATP or an electron carrier to drive those hydrogen ions against a concentration gradient. So these ion channels and pumps, they're very specific in transport systems, and they provide these pathways that really facilitate the control and the passage of different ions across this lipid membrane. So the ion channels, they provide this favorable passageway for these ions so that they're able to either diffuse across or be driven across that lipid layer to really drive that electrochemical potential. An example of that that we find on membranes, we can find in plants, and we can find it in chloroplast and the electron transport chains. We can find it in mitochondria. So this particular hydrogen pump, or proton pump rather, it receives its energy not from ATP like this image illustrates, but from an electron carrier. And so that energy that charge difference, that electric potential, drives those hydrogen ions across. So we end up with a concentration of high on this side of the membrane. And as those diffuse through that enzyme, that ATP synthase, which is a transmembrane protein, as those diffuse across, energy is transferred, and there are these excited electrons, energy is transferred, and where we phosphorylate an ADP into an ATP, and then we have more ATP charge. This is the part of the electron transport chain that occurs inside of that mitochondria. Now, this transport, it can also be coupled. As these proton pumps are driving hydrogen ions across, like we just talked about, there are these coupled transports where these transmembrane proteins can have binding sites with the two different molecules. And when the hydrogen ions bond to it, and in the example imaged here, sucrose, they will then diffuse across that membrane together, or that referred to as a co-transporter. So plant cells use the gradient of this hydrogen ion to generate these proton pumps and to really drive that active transport of a lot of nutrients into cells. So now these kinds of ion channels and proton pumps, they have really specific shapes with very specific receptors for the things that they bind to. And often they're observed in a variety of organisms. For instance, there's a whole family of these receptors, including the nicotinic acetylchlorine and serotonin type 3 and glycine receptors. They're all major players for human neurological communication, and they're also shared with bacteria and invertebrates and other vertebrates with these same receptors. So they belong to this family of these ligand-gated, and that some sort of outside thing binds to it to then change that shape, that three-dimensional model, in order for that structure to then change to drive those things across that membrane. So how does that interplay with the nervous system? Now, a neuron and a nerve cell is the fundamental unit of the nervous system, and having this structure and particular properties that allows it to really conduct some kind of signal or message. So it allows it to really talk to or deliver some sort of message, and it does it through an electrical charge moving across the plasma membrane. So going through some of the pieces that we have here, we've got the signal comes in, and it starts off here with the dendrites. And the dendrites are really, it starts at one of, or with many, usually, of these short, highly branched extensions of that neuron. And really what they're doing is receiving some sort of signal from another neuron or stimulus. And from there, that action potential travels to the cell body, where it's the part of the neuron that houses really the nucleus and most other organelles. And as that action potential travels, it then enters the axon as this charge difference. And the axon is it's typically like a long extension off of that cell body, and where it then enters these or where that impulse is traveling through, and it's going towards the target cell, where it's going to enter it's going to go through that axon, and as it goes down that cell line, there's this myelin sheath. So these are these specialized, specialized cells that wrap around it, and in between these, they're called nodes of Ranvier, where that action potentials are really generated between them, really passing that message on. And then when we get to the end of that neuron is where we come to the synapse, or the gap after the synaptic cleft. It's the junction, really, where the message crosses from one to the next. So a synapse, really, once we get to the end of that, is where we reach what's called a synapse. A synapse is really that junction, that space between the end of one neuron and the start of the next. So let's spend a moment examining that. So I'm really going to follow the flow of information here. So from the dendrite to the cell body to the axon. And there's a variety and there's an image here, sort of a graphic showing like as it travels down, but it's a one-way message, and there's a variety of different examples of this. And over here we have an example of a taste bud receptor. So we now know of a variety of 5 different taste sensations with sweet, sour, and bitter, salty. All of these, they arrive from, there is a whole diversity of flavors there through the of tastes in our taste buds, but this is one taste bud that can deliver any of these messages. So this it's associated with a component of this downstream signaling that occurs. This downstream signaling, where instead of that message or the mechanical mechanism being where through exocytosis, some sort of neuroreceptor or synaptic vesicle is expelled, this is actually a transmembrane protein. This particular one is the CALHM1 and the CLAHM3, where they're able to drive any of these molecules, any of these different flavors, these molecules stimulating that, deliver those across to connect to the receptors of the neuron, conducting as a neurotransmitter. So instead of functioning one way, there's actually essentially transmembrane proteins that are passing those messages through. So with our with the delivery of information, when an action potential, when that charge difference is traveling down that axon, when it reaches the end of this synaptic cleft, that presynaptic membrane, it encounters a region, or what's called the synaptic cleft, this space here, and it's really the space between them. It's a very small space, but they don't touch. That cleft is where these neuroreceptors are released out into, where they'll then bind with the postsynaptic membrane, or at least ligand-gated ion channels on it, that'll then open them up and allow ions to cross, then transferring that electric charge. Where with you can see that the sodium and potassium pass on, moving or continuing that particular action potential down the line. So we can get a little bit more specific in the different steps here. So first off, what happens is that we're going to go through these like step by step. The numbered steps here. In step 1, this is the action potential that has been traveling down that particular presynaptic neuron towards that axon terminal. Now what we're looking at is in the presynaptic neuron. And as that charge changes, then we've got calcium 2 ions that enter the synaptic node of the presynaptic axon terminal. What this does is it drives the charge up, which of this node, of this end, which ends up pushing these vesicles, these synaptic vesicles, towards the edge of the membrane. And when these phospholipid membranes bond with this outer membrane, these through exocytosis, here in step 3, these neurotransmitters are then released through exocytosis into that synaptic cleft, where then these little neurotransmitters, they're going to bind with those receptors, and these receptors are integral proteins that are crossing those membranes. And when these ligand-gated ones are open, it allows ions to pass through, and that's then delivering that particular that ion exchange is delivering that neuroreceptor, or that charge difference, where that action potential then continues down. So once an action potential reaches the end of that axon, then what? It then hops to the next dendrite and travels down, ultimately to the very end, either back communicating back with the nervous system, or delivering a message to the muscle, one way or another, depending on which direction that message is traveling. So to have for you guys is a real quick video review to sort of recap how this process works.
Our brains are constantly processing information. which requires communication between billions of nerve cells. Signals are passed from a sending neuron to a receiving neuron at a junction called a synapse. An action potential in the sending neuron travels down the axon until it reaches a synaptic terminal. The narrow gap between the synaptic terminal and the receiving neuron is called the synaptic cleft. The synaptic terminal of a sending neuron contains numerous vesicles filled with neurotransmitters, chemicals that carry information across the synaptic cleft. When an action potential reaches the synaptic terminal, the vesicles fuse with the plasma membrane of the sending neuron, releasing neurotransmitters into the synaptic cleft. The neurotransmitters affect the receiving neuron, changing the distribution of charge across its membrane. Let's take a closer look. An action potential is propagated down an axon by the opening and closing of sodium and potassium channels. When an action potential arrives at the synaptic terminal, it causes the opening of calcium channels, shown in green. Calcium ions enter the synaptic terminal through the calcium channels. Calcium ions bind to the vesicles containing neurotransmitters. This causes the vesicles to fuse with the plasma membrane of the sending neuron, releasing neurotransmitters into the synaptic cleft. The neurotransmitters diffuse across the synaptic cleft and bind to receptors in the plasma membrane of the receiving neuron. Here, the receptors are ion channels that open. Ions move across the membrane, changing the distribution of charge across the membrane. The neurotransmitters are quickly removed from the synaptic cleft, ending their effect on the receiving neuron. A single neuron can receive signals from many sending neurons. The signals arriving from the axon on the left are excitatory. They make the receiving neuron more likely to generate an action potential, as indicated by the green glows. Signals from the axon on the right are inhibitory. They make the receiving neuron less likely to generate an action potential, as indicated by the red glows. The two sets of signals cancel each other out, and no action potential is generated. Now, only excitatory signals are transmitted, and the receiving neuron generates an action potential down its axon. The signals that pass between these and countless other neurons constitute our thoughts and coordinate our activities.
So if you're interested in learning a little bit more about these action potentials and how it plays these structures and functions play a role in immunology and the synaptic cleft and also the neurotransmitters, here's a few sources you can go to. And these are the references that I used in putting this presentation together. I hope this review of ion channels and neuron communication was helpful. Thanks so much.